All right. Hi again, folks. Here we are. Going to wrap up this chapter on muscles. So as you look at page 352, we're dealing with muscle contraction. Now, what we've got to do is look at how a muscle is innervated so that it can contract. If you break the nerve, cut the nerve or something like that, damage it so that it can't transmit impulses to the muscle, muscle's not going to contract. Of course, you can stimulate it from the outside, like with an electric shock or something, but otherwise it's not going to work. So as you look on page 352, come down, this is module 10.4. Come down in the first column there and you see neuromuscular junction. That is one way of saying it. You can say the myoneural junction if you want to. Either way, just fine. Now, as you read through this, you're going to see terms, bold print terms, innervation, which means that nerve function is being attached to, in this case, a muscle. If we say if it's innervated, we mean it's got a nerve hook to it and it'll function. You see a motor neuron joins with a muscle fiber, and they call that junction, sometimes a synapse. Here we use the word junction, and synapse means a lot of times when two nerves come together, you got an impulse coming this way, and it jumps over to the next nerve and goes on to its terminal location, and they would call that junction a synapse. It's okay, no problem. It says the synapse of a motor neuron with a muscle fiber is known as the neuromuscular junction. So you keep reading there. It's got some real good stuff. It's real straightforward, not a lot of it though. The function of the neuromuscular junction is to transmit a signal called a nerve impulse. Now, we're going to say there's several ways to describe a nerve impulse. You can call it an action potential. You can call it an electrochemical message. You can call it a nerve signal, a chemical signal. There's a number of things that you could say that are all pretty much the same thing. So you've got the nerve impulse, which, as they say, is actually the action potential. We want it to come from the neuron, as you see the last couple of sentences, from the neuron, and get to the sarcolemma so that the muscle can contract. As you look up the top of the second column, you see it says the NMJ, which is just an abbreviation for neuromuscular junction. Got an axon terminal. If you'll look down at figure 10.12, you see the yellow bulb, and then you see that yellow tube going from it. That yellow tube coming toward the bulb is a telodendrium. It came from an axon. Remember, axons carry impulses away from the neuron body. So here you have the telodendria. You come down to this big structure that they call sometimes the synaptic bulb. And let's see what else they say. Got some vesicles inside there. You see the little circles. Remember how we talked about vesicles in chapter 3? See, this stuff never leaves you. That foundation material is going to be on and on and on. It's going to be carried over in the spring, after you get through with 210, or the summer, the fall, or whatever. It's going to be thrown at you again. And so inside the little synaptic vesicles, you see those little diamonds, those little kind of blue-green diamonds. Those represent a substance called acetylcholine. You can say acetylcholine if you want to. Some people say that. Most of us say acetylcholine, but it's not going to change the course of history. So anyway, the cell has to manufacture acetylcholine. So there's got to be some chemical metabolism. If you're manufacturing something, then you are, well, what kind of reaction would that be if you're making something? See how that goes all the way back. So don't forget to review. Now, you've got those vesicles with the neurotransmitter in it, and you see that the bulb does not touch the sarcolemma. There is a cleft in there. Now, a cleft is something we don't use too often in terms of just talking with each other. But if you've read some things in the Bible, you remember when Moses wanted to see God, God said, "Well, you can't look at you. Nobody can do that and live." So he, I think in that chapter in Exodus, it says that God put Moses in the cleft of the rock, put his hand over him. And so there's a cleft. That means there's a space in there. Put him in there for protection. So there's a space between the synaptic bulb and the sarcolemma. They are held in place by some connective tissue, but they don't touch each other. So there's a space in there, and that space has extracellular fluid. Now, you're looking at a muscle cell, that's why it's called a sarcolemma there, and then you've got a nerve cell. They're joining. That's a neuromuscular junction or a myoneural junction. And the space in there has extracellular fluid. So you got water, you got sodium, you got calcium and other things in that fluid, that extracellular fluid. And so if you can envision, the sodium is in that cleft. It's not on the inside of the sarcolemma, on the downside, in the muscle fiber, actually. So you've got sodium on the outside, you got some potassium on the inside, and so forth. And you have a charge there. You have a potential, a membrane potential. They'll tell you it's 70 millivolts. Some people say 85 millivolts. But anyway, it's charged, like we talked about just a little while back in the previous video. So you got these little proteins. You see the little purple things that are in the sarcolemma? Remember when we looked at chapter 3 and we looked at the membrane, we had all these proteins floating around in there. That's where they get the fluid mosaic presentation or theory. So you've got these proteins and their channels so that sodium can pass in. See, all that stuff from chapter 3 is coming up here now. Got to put it into play in terms of this myoneural junction. Excuse me, they sometimes call that sarcolemma. It's there under that synaptic bulb. They'll call it the motor end plate. So that's just another name for that particular part of the of that arrangement. Now, look over on page 353. And here we have, excuse me, a very similar picture. But as you look at figure 1013, you see this green arrow coming down the axon terminal. Nerve impulse. They call it an action potential. Somewhere down the line, they may use the word nerve impulse. So just be flexible, okay? It's all the same thing. So here comes that nerve impulse, and that's those positive ions going inside and stuff coming out. So you get negative on the inside, positive on the inside, excuse me, and negative on the outside. That's your action potential that's coming down to this terminal. When it gets there, it causes vesicles to fuse with the membrane, the phospholipid membrane of the neuron, and dump its neurotransmitter, which is acetylcholine, into the cleft. You see that the little diamonds hook on to the proteins. Those protein, when that acetylcholine hooks onto those, that protein opens up. Isn't this amazing stuff? And we don't even think about it twice. You just stuff that hamburger in your face. Me too. I do the same thing. And the muscles are working like that. Nerves are being activated. And all these impulses are happening. And we can even taste the good taste of the hamburger. It's just phenomenal how we're made. Just phenomenal. Anyway, they hook to that protein, and that protein opens, and you see sodium coming in. So the transmission has been successfully achieved. Now, let's go over to the next page. And you look at figure 1014. You see the sodium coming in, okay? Now, if you look to the left of the axon terminal of the synaptic bulb, see the little green arrow going to the left? Well, that means that impulse is going to be going to the left. And it's also going to the right. That action potential is going to the right. You can see, if you look a little bit to the right, along that green arrow, you see action potential. That's the nerve impulse. That's the movement of those ions into the muscle cell, into the cytoplasm of the muscle cell, and reversal of charge. That's the action potential. It's so simple, isn't it? What is that? But it's so important. So important. You take a drug that can stop that, and your muscles won't contract. And there are drugs like that. Anyway, you see that sodium come roaring in there? And so it's changing the polarity. It's reversing it. You see it's positive on the inside, negative on the outside. Whereas before it was positive on the outside, negative on the inside. Remember, that's what an action potential does. It changes the polarity. It reverses the potential. And so it's moving down the sarcolemma, and it comes to one of those transverse tubules that we talked about. The transverse tubule is an extension of the sarcolemma into the muscle fiber, the muscle cell. And so that impulse travels down that T tubule. As it travels down there, it causes the calcium gates. You see over here to the right, it says calcium +2 channel. Call it a gate, call it a channel. Either one's okay. And calcium comes out. Thousands of calcium ions. Drink your milk, eat your ice cream, eat your yogurt. And calcium comes out. All right? Now, look over on the next page. And I want you to look at the bottom picture. Excuse me, the left picture. It's got two parts to it. So calcium's coming out. Hadn't quite connected yet. But look at the little box. See the little box with the dots on it? See when they enlarge it? You see the purple grapes? Remember, that's the actin proteins. And the active sites of the actin proteins are covered by tropomyosin. And troponin is anchored also on the actin filament and the tropomyosin. And you see the head of one of those little myosin heads of the myosin molecules. Now, the calcium's coming out. Hadn't got there yet. Go to the right, follow the arrow, and you see in that big box, which is a magnified bit of the small box, you see the little green ball? That's representing calcium. And you see calcium attaches to the troponin. That's where number 1 is over there to the right. Calcium binds to the troponin, and the troponin moves, and when it moves the tropomyosin. And you see those little depressions in the actin? That's the active site. And that active site has got to combine with the reddish head of the myosin. Isn't this amazing how intricate this is? And yet, athletes, musicians, they move with such grace when they're playing a cello or a guitar or something like that, or they're diving in such a beautiful, a rhythmical way. Same way we're running. Some people just are beautiful running. And yet this is how it happens. It seems so cumbersome, but it works over and over and over again, just split seconds. So the head of the myosin attaches to the active site of actin. Let's go over to page 356. You see that picture up at the top. Yeah, I'm not going to hold you accountable for all of this, but you see what's happening. The head joins the actin site, active site of actin, and it pulls the actin in toward the M line. It's happening on the other side of the sarcomere too. See, so you got to pull in the actin into the middle. So the sarcomere shortens, the filaments don't, but the sarcomere overall shortens. Okay. And so you see down there, you got 1, 2, 3, and 4. I'm not going to ask you all about that. Just know that the heads of the myosin will attach to the active side of the actin, and they'll pull the actin in toward the M line. Look at the top of 357. I'll have to wait on that phone call. They'll have to wait. This is important. Look at the top of 357. There's a relaxed sarcomere. You see the M line. Now follow it down, and you'll see the myosin bridges, the heads pulling the actin in, and then you look down at the bottom and you see how the actin filaments have gotten closer to the M line. That's contraction. And it can happen so quickly. Think about the people with martial arts skills, how fast they can move their legs and feet, their hands. Just a blur. But you see what happens. The actin is pulled in toward the M line very quickly. And then on page 358, so we've got a contraction now, and I'm going to send out a video of that. I think I found one that's about 10 minutes long, but it'll say it in a different way that I say it. You know, I don't have a monopoly on how to say it, but it'll say it in different ways, and that's good for you. That's healthy for you to hear it in two different ways. Now, the muscle's got to relax. Because, you know, we can pick stuff up and we're holding it. I don't want to drop this thing, you know, and break it. Page 358, second column. A few steps here. Muscle relaxation. Come down the second line, has two components. Acetylcholine release stops. Remember when the message came down the nerve, it caused the release of acetylcholine. Now you stop that impulse. In other words, when you put whatever you're holding, your kitchen chair or something like that down, that message is not going down anymore to the muscle to pick up that kitchen chair and hold it. So acetylcholine release stops, and the remaining acetylcholine in the synaptic cleft, and sometimes even call it a gutter, synaptic gutter, is broken down by an enzyme. You look at number 1 under there, acetylcholinesterase. You got to say that 5 times every morning when you wake up. That'll get your lips and your teeth and your tongue in time with each other. If you see ASE on the end of it, what does that mean? It means it's an enzyme. And you know, as far as I know, you're going to say, it's a protein. You got proteins that are enzymes that sometimes break things down, sometimes catalyze the production of things, but you got proteins that are structural too, okay, and contractive. So you see in relaxation, acetylcholinesterase breaks down the remaining acetylcholine, and now repolarization. What brings about repolarization? Okay. Come down to number 2. The sarcolemma returns to its resting membrane potential. How'd that happen? That's repolarization. It goes back, it gets back where it was, positive on the outside, negative on the inside. Calcium ions are put back into the terminal cisternae. They're pumped back in, probably by a protein. Just start thinking like that, okay? And calcium concentration in the cytoplasm drops. Then you see number 4, troponin and tropomyosin go back to the shape they were before the contraction, and the tropomyosin covers the active sites of the actin, and the muscle relaxes. Think anybody ever asked you to write all those steps down? You better believe it. Okay? All right. Page 361. Just a couple of things. In order to have this contraction take place, we've got to have energy. So you look on 361. A little paragraph top talks about immediate sources of energy. And I want you to read that and know it. Creatine phosphate plus ADP, you end up with ATP plus creatine. The creatine's got a phosphate that can be added to ADP, which is adenosine diphosphate instead of tri. And so that's one of our sources of energy is creatine phosphate. And you see there's an enzyme that helps that. Notice that enzyme ends with an ASE, kinase. Okay? So there's one source. The other source I want you to know about is down at the bottom where you see glycolytic energy sources. What does that mean you're going to break down? How about glucose? Okay. So that first little paragraph, glycolysis, known as glycolytic or anaerobic catabolism. What are you doing if you're doing catabolism? Think about that. And you see at the last 2 lines, during this process, glucose is broken down to produce 2 ATP molecules per glucose. So there's where we get our energy. Okay? Now, let's go over to almost finished with this. Yep, that'll be okay. Yep, about 25, 28 minutes, we'll be done with this. Let's go to page 368. On page 368, you see in the second column a module 10.7. And you come down to where you see motor units. So you look in the paragraph and you see on the second line, a single motor neuron, along with its muscle fibers, which it innervates, connects to, is called a motor unit. Okay, makes sense, right? Muscle's got a couple of neurons in there on these fibers. These fibers attached to these nerve fibers attached to the muscle fibers. And those are motor units. Now, look at the bottom of the column. An average motor unit consists of 150 muscle fibers per nerve. Okay, but this number can vary depending on what part of the body we're talking about. Look at the picture on page 369. And you see a couple of neurons coming out. You see the red ones, and they innervate 3. 3 fibers. You look to the blue, and they got 5 fibers innervated with a different nerve. Okay, keep that in mind. Come down into that column underneath it. It says muscles. This is the second line that require fine control, like your fingers. You have a lot of nerves and fewer muscle fibers. Muscles that require fine control, such as those around the larynx or the fingers, will have multiple small motor units, often containing as few as 10 fibers per unit. So we have great control of these fingers. That's why we have to train them, and we can write, and we can make all these paintings and so forth. And there's a lot of things we can do with our fingers. Think about your lips and your tongue. Think about how you had to practice getting your lips and your tongue in just the right place. Sometimes we talk about the tongue touching the roof of your mouth when you use the word talk. When you say grass, you don't have that tongue up there, see? So we have to learn how to place our tongue against our teeth and also form a particular shape of our lips so that we get the right sound out. That's how we communicate with each other. Okay? Got a lot of control over your fingers and over your lips. It's not the case with your big old thigh muscles. They don't do delicate work. They get you out of trouble when you got to run away from a dog or something like that. So you get the picture there, okay? The more nerves and the fewer muscle fibers that are hooked to those nerves, the more control you have over a certain part of your body. So you can read about that. And in muscle tone, you see the terms in there on page 369. You got muscle tone. I'm sitting in my chair, and my torso muscles, they're not all bound up like this, but they're contracting with giving me tone, which keeps me upright as I sit in this chair. The muscles in my neck are doing the same thing, holding my head at a particular angle so that I can hopefully look it into my eyes to see this stuff. So muscles have this tone, which is just a little bit of contraction to, in this case, keep your head up. Hypotonia and hypertonia. You can read that and know it. Okay, last thing about the skeletal muscles for just a minute. Look on page 371. And you're looking at endurance training in the second column versus resistant training. Some of you have been athletes, maybe playing softball, volleyball, maybe ran track and played basketball, things like that. So as you come down to line 6, endurance training, what you're trying to do there is get your muscles, we would say, in shape so that you could run 2 miles as opposed to sprint 100 yards. Sprinters don't have as much endurance as a long distance runner, okay? So when you train for endurance, if you come down to line 6, excuse me, it says after endurance training, the muscle fiber has increased in the amount of enzymes, more mitochondria, more mitochondrial proteins, and a greater number of blood vessels. And so they can last longer in an event. One of the things the doctors usually want you to do is to, as you get older, you don't jog as much, maybe your joints are not as great, but you walk. And you try to walk about 4 miles an hour. gets the heart beating a little bit, and you're basically getting sort of an endurance training, which is good for you. Okay, muscles don't get real big. You're not into that, but you want endurance. If you're in resistance training, as you see at the bottom, then you're not looking for great endurance. You're looking for strength. You can pick up weight, push it over your head. Football players would do that. They would, because they want that. They have a burst of strength pushing the defensive man off or trying to tackle the guy. They don't run around trying to catch him for 10 minutes. So you lift weights in the resistance training. You see the word hypertrophy? Hyper, excess, trophy, growth. And so notice what happens. There, right, figure 1027b, it says you can see that both the numbers of myofibrils and the diameter of the muscle fiber increase. Fibers increase. So that's where you get bigger, bulkier muscles. It's good to have strength. You might not want to be as big as somebody, but you can look at the top up there, figure 1027, and see the distance and resistance and endurance training. And then down here, disuse. If you don't use it, you tend to lose it. It's like bone mass. We talked about that. So if you don't get any exercise, that's not good. You need some exercise. It's good for you. You don't have to become an Olympian, but some sort of exercise gets your muscles, maybe with some strength. But you can even lift weights with lighter weights, and you can do a number of repetitions instead of doing, say, 3 sets of 10 with a heavier weight, which is going to give you bulkier muscles. You could do, say, repetition with half the weight, and you would be getting your muscles somewhat strengthened, but also they would be strong in terms of lasting for a long time of some activity. If you don't do it, atrophy sets in. You start wasting away. Not good at all. Okay, I think the last thing we want to talk about is on page 373, and that's, you see it says smooth and cardiac muscle. We just want to mention visceral muscle. Okay, remember, smooth is a description of the appearance. Module 10.9 on 373, and just a couple of things here. As you look at the top of the column, and you see it talks about the difference between smooth and cardiac. Of course, you know the cardiac and visceral muscle are involuntary. You don't control that consciously as you do your skeletal muscles. Visceral muscle, they call it smooth, but that's just a descriptive term. But visceral muscle, you find in the viscera. It's in your GI tract. It's in your urinary bladder. It's in your uterus, if you're a girl. It's in your blood vessels. All of us have blood vessels. You don't have any control over that. And they're not striated muscles. They're what we call smooth, because the proteins, the myosin and the actin, don't organize themselves into bands. Okay? So, what functions? Peristalsis, that's the movement of your GI tract to push food through your tract, as well as to squish it up good, mix it with the enzymes so that you can break it down and absorb the good stuff and turn it into you. So you read about peristalsis, sphincters. How many sphincters do we have? That's a circular kind of muscle. One of them's right here. This is a sphincter. You got one at the other end too, don't you? We got some sphincters in between there. Got some in our urinary tract and so forth. And you see up at the top, they regulate the flow. In the second column, regulate the flow of, in our case, food through the GI tract. We also have some of those things in our lung, not in our bronchi, so that we can open our bronchi up if we need more air in, or we can close it down if we don't need it. You can read a little bit about the smooth muscle structure, just the first 2 or 3 lines. Smooth muscle They share the same proteins, myosin and actin, but they don't line up like that. So we don't have these striations. Okay. All right. That's the end of chapter 10. So I'll start looking on chapter 11 and get to work on that. But you guys can start on chapter 10. I will check over my Word document, try to avoid errors, spelling and all that, and give you guys a good example to follow. And I should shoot that this afternoon to you. Okay? You guys have a good time, and I hope you're going to knock out some good grades on the exam you're taking today or tomorrow. All right? See you later.